So nice to be here also today. It's my last lecture with you and you with me. Okay, that's obvious. So as I promised you yesterday, I would like to finish this last hour by presenting a part of the work we are currently doing in my lab that concern build of yeast cell factories in particular to produce some anti-cancer drugs that come from Catharanthus roseus, obviously. Okay, and in particular, we were interested in vincristine vinblastine, but we have managed two types of strategy. One strategy in the lab, we are alone to do that. And the second one needed a big consortium, a big European consortium in order to be conducted. So there is some perhaps redundancy because they are my slide of my conference for in 2 days, okay, in this congress. So I don't know if some of you plan to participate. I saw the program. It is a very, very good one. So, okay. And just 2 words for my lab. Okay. We are involved in 2 types of big area of research. The first one concerns the elucidation and also the characterization of metabolic pathway, not only alkaloids, even if it is historically one of the most important part of the work we have done in this area. But also we are now very interested by several polyphenols pathway, specific pathway, leading in particular also to specialized metabolite that have anti-cancer activity. We have lots of work with this type of molecules. And also we are interested in terpenes for several other reasons, in particular because we have nice contract with several cosmetic companies, and they are interested, you have seen already that, okay, by some of terpenes, and some of them are also interesting by this type of technology to improve also production of a specific molecule in particular. When I say the elucidation of pathway, I mean obviously identification of the genes that encode the enzymes that are responsible of each step of the pathway. But one of our major expertise is also to elucidate the architecture of the pathway, means to know where in the plant, okay, occur each step of the pathway. So at tissue level, we are looking at if it is in the epidermis, parenchyma, and whatever the type of tissue that you can find in a leaf or in a root, because you know that it is different, okay, between the organs. And when we have seen, for example, where are localized some of activities in tissue, we have also the idea to see where in the cell, okay, the enzyme are localized and the activity are also localized. This is a way also to find another intermediates necessary proteins such as transporter, to know effectively how the flux of the produced metabolite occur, okay, in the plant. All of these works take lots of part of the project we conduct in our lab. On the other side, we have developed for now, like 20 years, something like that, several biotechnological approaches, including in vitro culture plant or cell culture. And we do lots of things, perhaps quite similar as Professor Smita is doing also in her lab. So means to see if it is better to have vitroplan, inter vitro plan instead of cells in order to produce specific metabolite. Because what we observed for a long time is that cells are fantastic, but sometimes they can't produce the ultimate metabolites that are interesting for us. It was the case for Catharanthus roseus. We have seen yesterday that it is always the case. Now we know what, because the organization of the pathway is quite complex in the plants. And but now we have also developed another type of approaches, means plants, okay, vitro, some small bioreactors with a temporary immersion, okay, system. And we have found that in this condition, we may have very quickly a very high amount of biomass. And also we can act directly with the culture medium we propose to elicit sometimes this biomass in order to increase a lot, specifically the production of metabolites. So we have also developed this type of approaches, and all of these projects are collaborative with companies. We have a lot of collaboration with several types of companies. So this is concerned mainly this part, and also this classical, I will say that, biotechnology process that is also efficient. But what we have now developed from about more than 10 years old, but not so more, okay? It's all this new type of biotechnology that it is including synthetic biology, domains, that is production of plant metabolite in heterologous systems such as yeast cell. So we have developed lots of projects leading to the production of the specific metabolite, in particular in this type of cell. So I don't want to show you all the projects we have conducted, because some are finished, some are currently doing, some are in our mind, okay, for the future. So it's normal to anticipate every time what we want to develop. We need to be careful of what the market need also, because it is not necessary to develop this type of work if there is no issue at the end, if it is only the pleasure to do, okay? It's for scientists, it's okay, but for the lab, okay, it could be less okay, because we need to have lots of money to conduct all of this type of work. So, as you know, as I say to you, we are really concerned by anti-cancer molecule, because it's quite obvious that for the next years, unfortunately, okay, the number of people that could be concerned by this disease, and also by the death, because for some, it is the issue, okay? It is estimated at several millions people. And you can see here that, in particular, in Europe, North America, and Oceania, okay, the number of people that are concerned by this disease is really increasing a lot, perhaps less here in Asia, but I think you are concerned as us, okay, at the same level. So now we know that there is a lot of molecules such as antibody that have been also introduced in the different treatment against this disease. So we are also working on a yeast that will be able to produce some antibodies, and some antibodies linked with ADC, which is a way to to orient the antibodies inside the cells. But on the other side, we know that there are still some plant molecules that have a very high efficiency against several types of cancer. So we develop also production of these molecules, okay, in yeast. Mainly, we work on 3 types of molecules, and all of them are concerned one of the global classes of metabolites, terpenes. In particular, we have started to work also on taxols. Not on artemisinin, we have stopped, because there is other teams that are working currently on, and it's not necessary to be always in competition, okay? And we are mainly concerned today by producing podophyllitoxin. Okay, I already spoke about this to you. We have now very nice results. It's a secret, because we are waiting for a patent, okay, to work with one pharmaceutical companies. And obviously, we have developed several works on producing, in particular, or vinblastine or vincristine, or their precursor, as I will show you now. We are also now working currently on different molecules that are of high interest for cosmetics. I can't say nothing about that because it's also some secret. Perhaps future years I could come back and explain you what we are doing. So the question is always the same. These plants are mainly cultivated in fields, okay? That could have some limits. For example, we know that some climatic events could have a very bad impact on the culture. So for 1 year, you may have some problem for having enough raw material. There is also, we have seen that with very recently, okay, you have also different geopolitical conflicts that lead to difficulty to have access to this raw material. Also, what it is happened with the podophyllum problem of plants that are rare, difficult to cultivate. So all of this they have some consequence is the increase of the field, okay, to cultivate these plants. But the second consequence of this is obviously problem with deforestation, because to increase the surface of culture, we need to eliminate a part of the forest surface, which is becoming a very high problem everywhere. And also there is a higher competition with plants for food. Obviously, we have increased our population number, so we need to have more foods, okay, to produce, not only for us, but also for animals, okay, that are also our food that they have. So by this way, it was interesting to develop a specific biotechnological process for producing this type of molecule, because obviously we have observed for several years that some of very important pharmaceutical molecules are shortage, okay, for several. It is the case for several years, and in particular here you can see that the two most important molecules that are missing every years at what moment of the years is vincristine and vinblastine, and also this one, etoposide, that come from podophyllum. And in the 10 first molecules that are missing on the market at what moment during the year, at least 3 are plant molecules, plant metabolites. So now it's important, it's a real challenge to find alternative way to produce this molecule instead of increased surface of cultivation of the plant that are at the origin of this metabolite. Okay, so the concept you know now, it's exactly the same problem, the same thing I have explained you before, just to see that here I will show you the main project we have conducted using this strategy in particular, means not the neosynthesis of the molecules from the sugar to the last molecule, but we have managed at the beginning, because it was the first year we have started to work in this type of area. So we wanted to test our ability to conduct this type of project. It was also a moment where I have recruited a specific scientific expertise in order to be sure to conduct in a well manner this project. So we have started by this bioconversion strategy. So the principle, you know already that it is always the same workflow. This is the workflow we have established in the lab. This picture comes from a review we have recently published. This is why I have always the same. Okay, so this step was okay. This step we have generated so many omics data. Okay, and we have also worked with several companies or several laboratories that are mainly involved in the analysis of this data. And I have also recruited 2 assistant professors that are specialists of this type of work, because I was not able to do that by myself. It's not my own expertise. So I have increased the lab team with this new competence. And with them, we have find lots of new genes, okay, and we have started to build this particular cells, engineered cells. But yeast is not plant cell, so we are expert in plant cell, not in yeast cell. Even if we can do molecular works, I mean transformation, genetic transformation, this is quite what we can do because it is our expertise. But we have no idea how to manage the culture of these cells in order to have the high efficiency of their biomass. So we have also recruited a new assistant professor that is a specialist in yeast, because there is also several manners, not only engineered manners, okay, to push this type of cells to express specifically some character. So there is also some other theory that's are very interesting, say to push some to put these cells in a specific under specific stress, environmental stress, and this could be an accelerator, okay, of a specific adaptation that could lead to modification of metabolisms too. So the expert in yeast we have recruited help us also to manage different things on this type of cells. And now we start to become a little bit more expert, okay, in yeast cells than in plant cells. And the last point was also very important, even at the lab scale, because when you are talking with industrial companies, they say to you, okay, but you have these cells, but what I can do with? I don't know. Yes, you have to know, because in fact, several times they are interested by your results when you arrive to the first production in bioreactor, and not only at the lab scale of the bioreactor, a small one with 2, 3, or until 5 liters, but you need to go at least at the pilot, okay, phase, okay, if you want to be interesting for companies. So this is why we have done lots of projects to have money, okay, in order to buy several type of bioreactor from about 2 liters until 50 liters, in order to have the possibility to scale up by ourselves the first phase, okay, of production of this engineered yeast, in order to have the possibility to discuss really with company in order to transfer as we are done, okay, directly to the company. At this moment, we are in discussion with 2, okay, in order to transfer some of our projects to them. So you see, it's not so easy. It needed about 10 years, okay, to establish this workflow in my lab, to have time to recruit people that are expert to the different discipline. Whatever now there is a new discipline, for example, how to manage artificial intelligence, to make some machine learning in order to have a better efficiency at this type. We have collaborators for that. It's not necessary all the time to recruit people, because sometimes it needs not only one person, but a team, okay, to have access to the information. And also we are working with people that are more involved, okay, in this bioprocess. We have recruited also one, okay, that is an expert in bioprocess systems. So it is very nice now, because inside the team, we have discussion of about all the steps of this type of workflow, and we are able, in fact, to understand what are the problems at each moment and what can be the solution, if we can find some in the team, or if we need to have new collaboration in order to have a better, success at each step. Just to show you that it's quite complex, in fact, to implement this type of work in a lab that was not a specialized lab, okay, in this type of work. It needed for us about 15 years. So I come again on the Catharanthus roseus, and as you know, Catharanthus roseus is able to produce 2 alkaloids, vinca alkaloids, catharanthine and vindolin. And these two alkaloids are precursors of, in particular, vinblastine and vincristine. What I showed you yesterday is that, in fact, companies such as Roche and Roth are interested by extraction of these two precursors from the raw material of Catharanthus roseus, because they make by semisynthesis the final product, which is more interesting because these two molecules are in a very high amount of concentration compared to the last one, vincristine and vinblastine. So what was the project we developed, we proposed at the beginning? It is a project that was called BioProfarm project. It takes part of a big call from our regions, because the region where I am living is very interested and it's very helpful for research when there is some really important issue. So we proposed them to develop two lines, okay, of yeast. The first one will be able to produce the first precursor, okay, and the second one was also be able to produce the second precursor. So in this project, we have this particular company as a collaborator. It's a big company, an international one, but they are actually concerned by chemical synthesis or extraction, okay, not by this type of work, and they were very interested by implement this type of new technology in their own company. So they accept to participate to our work, and you will see why. Because in fact, the strategy we have started at this moment was not to, as I say to you, not to start with sugar, but to start with a precursor. For example, here, strictozidine, okay, that can lead directly to the production of catharanthine. You can see all the nice, okay, metabolic pathway that have been presented to you by people that are concerned about this subject. So you can see here that it's needed about 7, 8, I don't remember exactly the number of the steps, okay, that occur between strictozidine, the first precursor to catharanthine. But it is also the case for vindoline. One of the first precursor of the specific branch that lead to vindoline is tabersonine. So the result I want to show you today concern in particular, okay, this pathway. It will be exactly the same scheme we have conducted for the second one. So we have developed a strategy of bioconversion, which is a very simple, okay, instead of the neosynthesis. So I have already shown you this picture. This is the total metabolic pathway that have been elucidated by our lab. It is exactly what I showed to you. We have found that at least 3 different tissues are involved in this metabolic pathway. And also, for example, in this tissue, that it is an internal parenchyma that are near the vessel, okay? And in the cell, all the steps occur in at least 3 or 4 different organelles. It starts in the plast, okay, with the production of the IPP and the MAPP by the MEP pathway. Here they are. After this precursor are taken in charge by several enzymes, in particular this one, geraniol synthase, to produce one of the first precursors, geraniol. This precursor is after taken charge by 3 different enzymes, you see, that are linked to the reticulum endoplasmic in order to produce one intermediate, loganic acid. And this intermediate is not accumulated in this type of cells, is, okay, transferred to another type of cells, the leaf cells. And in the leaf cells, this loganic acid is also taken charge by several enzymes that are, most of them are CYP proteins, so they need to be anchored specifically, for example, here to the reticulum endoplasmic membrane. Some are also in the cytosol, and there is at least I think 7 steps in which that lead to produce an intermediate that is directly transported to latticifere or idioblast. Latticifere and idioblast are very specific cells that are in the parenchyma. Okay, only one cell. Okay, it's not a group of cells. And in this cell, you have the 2 last enzyme, acyltransferase, that and D4H, that are able to condense, vandoline and catarantine to produce vinblastine and vincristine. Okay, you see, it's a very complex one. And it was interesting to have this view, before starting this work. So we decided to produce vindoline by feeding some yeast cell with the tabersonine, precursor. And you see in the plants, all of these steps that start with this particular hydroxylase, T16. H, to that, the last one, occur normally in 2 types of cell. That was interesting to also take in mind. And also, all of these enzymes are not always linked to a membrane. They can be at the beginning. They have some anchoring on membrane, but the last one are more in the cytosol of the cell. So it was interesting to know that, because technically, it is quite easy to imagine to transfer all of these 7 genes, because there is 7, in the genome of the yeast cell. And why we have a very nice collaboration with Accentis, it is because this company is a leader for the production of tabersonine. They used to extract this tabersonine from seeds of an African tree, and there is no limitation to have this access to this precursor. So even the idea was, if we succeed in this production, it will be easy for them, to implement the process, but also to have access to the first to the molecule needed to give to the yeast. So it was a nice configuration. It is always. So what we have done? We have done several things. Here is quite the last one. Firstly, we have started with epidermal plasmid. We have discussed about this yesterday. And we succeeded to have some nice bioconversion, not of all, because we have work block by block, But what we observed is that it was really unstable, because depending of the cells, we lost, in fact, some of the genes. So we have quickly shift to an integrative strategy, means to introduce the genes directly in the genome, of the yeast. For that, there is 16 genes, chromosomes, chromosome in the yeast, and in some of them, in particular in a chromosome 12, we have found that there is a specific organization of the genome. In particular, it exists, several genes. Here they are proposed in red, They are very essential for the life of the yeast. For this purpose, these genes are highly expressed, in the cells. So means that between these genes, it exists some specific site sequence that have been qualified at hotspot, because if you are able to insert a gene in this specific sequence that are not a gene, so you have no effect on the genome, of the yeast, but you are in an area where the transcription is very, very, very efficient. So by this way, not only you have to choose a promoter that could be interesting to stimulate the transcription of the gene, but you are also in a region of the genome where you are sure that this gene will be expressed naturally in a very efficient manner. And as you can see, there is not only one hotspot, but there is a lots of hotspots that could permit to imagine to introduce not only one gene or two genes, but a large number of genes, okay, by this way. Obviously, we have used the CRISPR-Cas9 techniques because we wanted to target specifically the position of this hotspot in order to be sure to integrate the genes in the good manner, and also to be more efficient and to decrease the different transformation we have to do. We have create specific bidirectional promoter, means that this promoter will be able to stimulate the transcription of two genes at the same time, at the same amount, okay? And so we have created some specific on va dire molecular construction between new promoters and between different genes. And if you put some together on the same cassette, you can introduce in one time at least 4 genes, for example, at the same time. But we have done this 2 by 2. The idea was to have a genomic context with a high transcriptional activity. It is what I explained to you. It is also to guarantee the genetic stability for the further generation of the cells, and also to using CRISPR-Cas9 technology, we don't need selection markers for this. So this was also very interesting because there is a lot of markers, but the number of markers is quite limited. So if you want to implement a high number of genes at one moment, it could be a difficulty. So by this way, as we don't need some, it is quite easy, to implement a large number of genes. And here, these promoters gave the advantages that the expression is constitutively strong. That's good. That is okay. So this is what I have already shown to you. So what we have done, we have implemented, this is my yeast, and we have put all the genes, the 7 genes, from T16 hash. We have several isomers of a gene. We have chosen one of them that seems to be the more the more important one also in the plant. So this is why there is a number 2 at the end, And also we have implemented all the other genes until that. And what we observe is this. When we have built this first yeast, and if we gave to this yeast some tabersonine, we are able to produce some vandoline. We were very happy. But the problem was we were also able to produce this metabolite, vandorosine. And vandorosine is a metabolite that it is not possible to convert in vandoline. We have so it's not a desiderate, metabolite. And this metabolite was mainly produced, and not a vandoline. So there is a very excessive production of this molecule that was not expected, and that was not, it was not interesting for us. So what can we do? Because why? We have tried to understand why this molecule is produced in the cell. So what we have observed is that this enzyme in particular is also able to be linked to the tabersonine. It is what I have explained to you, the promiscuity, of enzyme activity. And when this enzyme take this molecule as the first one instead of the product of the methyltransferase, in fact, it is not exactly even the same product that it is produced at the end by using exactly the same sequence of the branch pathway. So you see, it's also a way to produce several other non-natural protein molecules by a natural way. this is a derivative, okay, that it's not present normally at or very, 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 very low amount in the plants. So how can we do to try to avoid, okay, this particular branch? The idea was to increase the number of copy, okay, of the genes that are the first two steps, okay, of this metabolic pathway in order to, increase the flux of tabersonin to the hydroxylase instead of this T3O. So we have at least double the number of the copy of the gene. It is what we have shown here. And in this condition, what happens? It happens that no vanderosin was produced. Okay, means that this pathway is not in fact chosen for by the cells. And we have increased the production of vindoline, but we observe also a very high concentration of this molecule, this acetoxin vandoline, that it is the product of these particular enzymes. What means? It means that in fact here, the capacity of this protein is less efficient than this one enzyme. So it means that here, time for transforming this precursor in this product is longer, okay, than the time needed to produce by this way these intermediates. So what will be the solution? We have tried to change, for example, some of this enzyme, but there is not a lot of there is no isomer, okay, and there is not a lot of possibility to find a new enzyme that could be able to do this transformation. So we have done exactly the same strategy as here. We have increased the number of copy of the gene that encode these specific enzymes, and by this time we have put three copy of this gene. It is what it is shown here. But in this condition, as all the acyltransferase you could see in my later need to have some cofactor, we were obliged also to increase the different pathways that are responsible to produce the cofactor needed for the DAT or for the D4H hydroxylase enzymes. And by this way, we obtain this, okay? This is before, okay, the augmentation, the increase of the last gene of the pathway. And here are the results by using this strategy of three copy of the last genes. And what you can observe is here at several hours after to have feed the cells, the yeast cell with the tabersonin. Here we can see the first production of the first, okay, intermediates that accumulate, including desacetoxyvandoline. But 2 days after, we can see that this acetoxyvandoline starts to decrease a lot, and we can observe here the production of a high level of vindoline, more than in this case. And after 3 days, 4 days, not 3, 4 days. We can observe here a very interesting level of bioconversion from tabersonin to vindoline. So this was one of the strategies we can do in order to have a functional branch, but it is not the only one. It is just in this case, the strategy is to increase the copy number of genes. What we have tried also is to increase more, to put 4 and 5 copy. And in fact, at this moment, it is not increasing, but it can be the contrary. We can observe a decrease of production because, in fact, there is some other secondary effect of this high number of copy of the gene that could lead not to the good answer we want. So by this way, we have stopped, okay, with this gene in 3 copies. So what it is interesting here, it's not exactly the amount, it's the level of bioconversion. 77 is a very, very efficient, okay, number of conversion, because in fact, depends of how many tabersonin you give at the beginning, you will be able to transform 77% of this tabersonin in vendoline. This starts to be a very important criterion for the company to say, okay, we are interested by this. But at this level, we were in flask, not in bioreactor. So it was important to see if we transfer these cells in a more, in another process of culture, it could be exactly the same type of the result. So we have put them in a bioreactor just here to see that we have also optimized several other abiotic conditions of the culture, in particular the pH. We have made lots of experience by increasing or decreasing the pH step by step, okay? And we have found that some of pH, we were able to increase this level of bioconversion up 85%, which is very, very, very interesting. And by this way, we have optimized at flask level several parameters. So after that, we scale up this in a first small bioreactor of 2 liters. We have some, now we have 4 in the lab, okay? And what happens is that, okay, we have tried to different process. I don't want to show all the experiments that have been done, at least 2. Here you have 2 experiments. We put some tabersonin by a pulse, okay? It's not a continuous feeding of tabersonin. It is given at what moment in particular. Here it's in blue. Okay, you have 1 or you have 2 pulse, depend of. And here you can follow production of vindoline in red. Here are the other type of, and what it is also interesting is to see when we give some nutrients to the cells, you know that very well, okay? We need to see if we do pulse nutrient or debit nutrients, means we give some nutrient along the time. And you see here in this experiment, whatever the type of nutrition, okay, or by pulse or by debit, we are able to produce the same amount of tabersonin. Means that in this case, we have converted less than 80%. Okay, because this was the quantity of tabersonin we give at the beginning, and these are the quantity of vendoline obtained after these 2 type of. But what it is interesting is that continuous feeding seems to be less effective, because we can see that at what moment it starts to decrease. Okay, so we decide to and also there is also some accumulation of intermediates that could be not interesting. So this is why after we have, it's the contrary. I made something here. When you have this type of nutrition, you see you have an accumulation of intermediates. This is intermediates that are not present when you have a debit nutrients. So we have maintained this type of system to give the nutrient. And what we have done is we have increased, okay, the tabersonin content at the beginning to see if we can have also something that could be interesting. And what you see here is that when we give to these cells higher concentration of tabersonin, we increase a lot, okay, the probability to produce vindoline up to 161 or 266, okay, of vindoline. And here it's the maximum conversion percent we observe even in bioreactors of 88%. That was very, very important because it was, I think, the first time it was shown the possibility to produce vindoline by a yeast, okay, up this rate of bioconversion that have been published, okay, in this journal, and it is perhaps the best vindoline production report to date until today. Here are the two bioreactors we have in the lab. So What happens now? As we have opportunity to have access to 50 liters bioreactors, what we have started to do is to transfer this to culture at a low scale to a bigger one. And what we need to increase, obviously, the quantity of tabersonin. This is why we are very interested by the collaboration with Accentis that is able to give us a large amount of these molecules free. So it's interesting for that. And by this way, we have started several first experience. Firstly, to see how they grow without any tabersonin and something. We need to see the possibility to have a high biomass in this particular condition. And it seems to be okay. So the next step, I think it will be in the next few months, will be to implement these cells with tabersonin in order to see if they are always able to bioconvert this molecule in vanillin and also with the same rate, okay, of bioconversion. If it will be a success, I'm sure of that, okay, we will be ready to discuss with the company to know how to transfer all the process in their company. So we are lucky because, in fact, in Tours, there is a new institute named BioCube Institute, in which there is a lot of facilities, including this bioreactor. We have partially participate to the financial support to acquire this type of material. This is why we are quite, it's easy for us to have access to this type of material. But behind this material, there is also different other facilities, such as UPLC, in order to follow the production of the different molecules during the process, because we need to take some samples every time to see what happens exactly in the cells and so on. So what I don't want to show you today, it's we have exactly do the same with catarantine, production of catarantine, but the difference was quite big, because when we have started the project, we all the genes encoding the branches between tabersonin and vanillin were known. Okay, we had already identified all the genes, so we have start immediately. Okay, the building of the yeast, but it was not the case for catarantine. So we have spent at least 2 years and a half in order to complete the metabolic pathway that is between streptozinine and catarantine. And what we observe is that it was a too long one, so we have tried to shorten this pathway for the bioconversion strategy. And we have work in this case with the chemists in order to ask them, instead of giving some streptozidine, we wanted to give an intermediate that it is very difficult to obtain. So we have made a specific collaboration with the chemist in order they can produce for us, okay, this intermediate, stemmadine. And it was a success too, because for her it was interesting, because nobody before, no chemist before have established the protocol to produce this particular molecule. And she produced this molecule in a very sustainable manner, in order to be always in the same meaning. So now we can have, we have, as we had for vanillin, all the tools, okay, in order to build this new strain. Now we have a first prototype that are able to produce some catarantine, so it's nice. We will have time to optimize this new strain, and after that, it could be very interesting for the society, for the companies, the Accentis, because they will be able to produce both, okay, precursor, and they could sell, okay, this precursor, for example, to Roche or to different other companies to produce the final vincristine and vinblastine. So it is a very nice project. We started 6 years ago, something like that. So it was not so long to have this first success, but it is not finished, as you have seen. We have different other steps to do, but we are currently finishing them, and it will be perhaps a good success story in some years. But what happens in parallel, because as we have published several papers concerning this work, we have been contacted by two people, Michael Jensen and J.K. Sling. J.K. Sling is the father, okay, of this technology. He works for Ameris, okay, and he was involved in the first farnesen, beta-farnesen and squalene, okay, yeast able to produce this molecule. So he has a very large, okay, influence in this domain, and both of them contact us, and in particular, one of my colleagues of the laboratory, Professor Vincent Courdavot. I was the co-leader with him of the previous works, and he was contacted for this, and the idea was to make a consortium, a European consortium with them. Michael Jensen is at the Denmark VTU Technical University, so it's a famous one, okay, and Jeff Klisling is one of his collaborators. He's not in Denmark. He is from DTU also, but also to different other companies. He is everywhere, okay? And the idea was, can we manage, okay, a yeast that could be able to produce vinblastine, okay, by a neosynthesis way? Means we want just to give some sugar, okay, and they will be able to produce strictozidine, that is the first intermediate of this metabolic pathway, and this strictozidine will be able to be transformed in tabersoline and in catarantine and tabersoline to vendoline in order to produce vinblastine. That was the project. The project has received financial support from Horizon 2020 of the European Hockey Association. It was a project of 6.8 million euros, so it was a very big one. We were not only 3, okay, we were 10 collaborators. In these 10 collaborators, Maxentis was also the companies that could be also interested by this technology. But there were also other companies, and in particular 2 that are involved in developing some software, some system of analysis of data, and another one that was specialized in generating new omics data. So because in this case we have developed lots of genomic data, and our work in this consortium was to identify new genes in case of it was necessary, yes, and also to participate to the construction, okay, of the yeast, the genetically modified yeast, and the result was very good. It was a paper that had been published in 2 years. 2 years ago, it was at the end of the project. The project was for 4 years, only 4 years, okay. And by this way, what was interesting is that during this time we have discovered so many enzymes that missing, okay, in this metabolic pathway. This is very important because it is now perhaps one of the most well-known metabolic pathway complex with about 30 different steps from the beginning to the end. All of these enzymes have been characterized at enzymatic level, cell level, organism level, okay, so to complete completely the view of the architecture of the pathway. And what it has been done is that, in fact, the building of the prototype cells have been done step by step, block by block. Okay, so the first one was to implement the first gene leading to strictozidine production. So it was the first time that we published yeast able to produce strictozidine. And you see it was about, I don't know, perhaps 10 or more than 12 genes that have been implemented at this time. After, from this yeast, all the consortium, okay, have added another blocks leading to the production of catarantine, for example, okay, and tabersoline too. So it was a success again. We will be able to produce both of the two precursors, okay, in the same yeast. After that, it has been implemented, you see, the pathway of Vindolin, okay, the same pathway we have before implement in the first yeast, and also the several other steps that lead to the production of beta-carotene here it was. So what was interesting is that at this moment, here are the different type of the genes that have been implemented at the first type to wild type to the first intermediate that able to produce strictozidine. Here we have added several other genes to produce tabersonin. De novo means it was with sugar, not with anything else. And after here, we were able to produce also Vindolin and tabersonin, okay? And at the end, we have this and this cells that is able to produce Vindolin and catarantine. The only step that it is missing until now is the condensation between catarantine and Vindolin to produce vincristine or vinblastine. But now it's okay. At this moment, it was not exactly well done, but it is okay. What I don't show you here, it is the ratio, the yield of production that are low. That's true. But what it was interesting for us is that, in fact, the job have been done because the objective was to propose a prototype of a yeast cell that will be able to produce this molecule. And we have introduced more than 30 genes in these cells in order to manage the production of this metabolic plant metabolites. And what it is nice today is that Michael Jensen have taken the time to create his own startup in order to continue with us, okay? The optimization of the culture in bioreactor is this type of enzyme, the yeast. But during all of this step, we have created lots of collection of other type of yeast that are able to produce several intermediates that are of very big interest for several other applications, or to be a precursor for other type of metabolites, particularly metabolites that are produced in roots, like nericin, for example, and so on. So we use all of this material to develop now several other type of cells that could be in a few years, I hope, that could be used to produce several type of vinca alkaloids, intermediate vinca alkaloids. Okay, so it is our nice story. I wanted to share this with you to show that it can be very simple as to produce farnesene, but it can be more complex, but not impossible. Okay, if we want to produce several things. I want to thank here all my teams. Okay, it's a large part of my teams here. All of them are professor, or like Vincent, and also assistant professor. There is some engineer here, and also a PhD that have done a very fantastic works during this time. We, with the different contract, we have recruited lots of postdoctorants. Here are some. Okay, and we have also a very nice collaboration for quite 10 years with Professor Sarah O'Connor from Jena University at Max Planck. Okay, she was in our lab 2 months ago. Okay, we have a very close collaboration with her, in particular Vincent and her, and also with now Michael Jensen, and we continue to work with him, and also indirectly with Kay Kissling. Okay, to continuing this type of work. So yes, conclusion, this is what we are doing. We have published recently a new review in which we have summarized all the metabolites, the plant metabolites that now able to be produced in yeast over the last year. So it's quite recent. You can see alkaloids. Okay, there is not only ashmalicin, Vindolin that can be produced, but we have some other that are able to produce noscapine, and also different other tropins that come from other type of plants. You see here, scopolamine, we have already spoke about this. There is also some of cells that have been built to produce several terpenes. Okay, here are the list. Not only farnesen or limonene and so, there can be more complex one, and some also phenylpropanoids or beta-alanines. We don't have spoke about beta-alanines, but resveratrol is part of this type of terpenes. And you have seen yesterday that there is several system to produce resveratrol. Okay, and there is some nice also in yeast. Okay, so to conclude, it is here some remarks. I think you will be able to do exactly the same without me. Okay, that microorganisms are useful system as green factories for production of this type of molecule that could be used for food, pharmaceutical, and cosmetic products. We can observe, in particular, I don't speak about that, but in the food domains, there is an increased interest for this type of methodology. Okay. What it is interesting is that you have seen we have not finished to optimize, okay, this type of systems in order to have a higher production and a higher quality of production, whatever the season, whatever the plant, whatever all the environmental, parameters. We use a very small number of yeast, 3 you have seen, but there is so many others. So at the lab scale, we can also screen different other systems that could be more efficient for the future, for example. We are also looking at some new biological tools and technical tools, as it was, for example, with CRISPR-Cas9. We don't know, perhaps new tools would be developed in the future to be more efficient also. And it starts to be to have some successful commercialization, but not so much at this moment. So we are completely confident that it will be not the case. It will be increased, okay, the number of this commercialization in the future. I just want to share with you also 2 other slides. It was nice. It was for another lecture I gave in Poland, for example. They were more interested by food and also cosmeticals. And here it's nice to see that in different way of application, traditional fermentation, enhanced fermentation, animal feed, and so on and so on. The steady start of what it is occurring is what you know, okay? But here, there is a lot of projects that are based on engineering, okay, specific cells in order to produce specific molecules, yes, but also specific yeast lines to optimize also traditional fermentation way, for example, for beverage and so on, and also to fermentation for producing different other systems. And in particular, there is also lots of works that at experimental stage, okay, it's in lab, that concern the use of single cell to produce obviously proteins. And what it was nice is that, in fact, in this paper, they explain the possible platform that could be imagined. For the moment, it is not the case. It is a projection for the future, where with specific software, okay, and you can design what are the molecules necessary to have, for example, some flavor, some odor, some taste, okay. And after, with intelligent bioreactor, you could produce before, okay, several engineered yeast that have included the genes necessary for this type of purpose, okay, carbon assimilation, biofortification, it is always interesting to have, okay, and also color and flavor, for example, is here of meat. Okay, and after that, you can propose production of this enzyme. And what was original in this paper, you can take a look of this, is that you can associate this system with a microbial 3D printed, in order to recreate according different, you can imagine here, lots of number of bioreactors that could, make something similar to meat, to bread, to tomatoes, to different things. And you can be able to reconstitute, here an hamburger, it's quite symbolic, to produce, but it could be it is already the case. We can make some artificial meat by this type of work, so it's not completely crazy, to imagine that. So, but the idea is to use a specific engineered yeast, to do the raw material, molecular material, to build this type of things. And they have seen moreover. No, sorry, I forget. No, it was in the title, because this was imagine for space nutrition in the future station for the colony, the human colony in the space, or on the moon perhaps at the beginning, but also on other planets. I discussed this time with some people that are actually here from Cosmos or from this disciplinary. It was not so crazy too. They are thinking about several systems to facilitate after the life of people in the space. And the question of nutrient is always important, because people need to eat. So how to lead to continuously production of food. I show you that the indoor farming are always in this meaning too. So perhaps it could be not only one way, but several ways that could be tried, for this future colony in the space. I think I will be not here, but it's too much. I will be interested to see that, but it's nice to see that we can imagine how to transfer all this technology for several other applications in the future. So this is exactly what I want to finish. We have now the proof that this platform, microbial platform, could be very interested to continue to develop, whatever the domain in which it will be the case. And what it is, let's turn on to all of this advance in metabolic engineering that will have a strong impact, I'm sure of that, in our future. So thanks a lot for your attention. It was a pleasure for me to go here. So you have now a part of the vision of what we are doing currently in our lab. Thanks.